Hey everybody, we are going to move on to chapter 15 and we're going to look at the chromosomal basis of inheritance. So we've talked a lot about Mendel. His paper came out in 1866. It wasn't until 1875 that mitosis had been worked out, and then in the 1890s, meiosis. And then in 1902, we had Sutton connect chromosomes to meiosis. So he developed the chromosome theory of inheritance and said that Mendelian factors or alleles are located on chromosomes, and then it's going to be the chromosomes that segregate and show independent assortment. So again, this is just sort of the diagram showing all of that. Then we have Morgan, and he chose to use fruit flies as a test organism in genetics. And this allowed the first tracing of traits to specific chromosomes. So Drosophila melanogaster, the fruit fly, was the early test organism for genetic studies. And fruit flies are still used for a variety of reasons. So they're small, they're cheap, very short generation time, they produce lots of offspring, and they have few chromosomes. So the genetic symbols that you're used to that Mendel used were the upper and lowercase letters. So example, if tall is dominant, then it would get a capital T, and then short would get a lowercase t. Morgan used slightly different symbols. So he used a symbol from the mutant phenotype and then a plus sign to indicate the wild phenotype or the wild type. So an example here would be if you have a recessive mutation, which would be white eyes, then W would be white eyes, lowercase w, and then W plus would mean red eyes, which is going to be the wild type. Then if you have a dominant mutation, here we have capital CY, which means curly wings, so that is going to be the dominant mutation, and then CY plus would be normal wings. So Morgan observed that a male fruit fly had a mutation for white eyes, and so he crossed that white-eyed male with a normal red-eyed female, and the F1 offspring all had red eyes. So based on what you guys have learned so far, what type of eyes do you think white eyes are? Is it dominant? Is it recessive? It is recessive. All right. So then he crossed two members of the F1 generation to get the F2 generation. He expected the F2 generation to have a 3-to-1 ratio of red to white because the white appeared to be recessive. He did get this ratio. However, all of the white-eyed flies were male. So he decided or determined that eye color trait appeared to be linked to sex. And so he is responsible for discovering sex-linked traits, and these are genetic traits whose expressions are dependent on the sex of the individual. So here we have depictions of the fruit fly chromosomes. So we have the female right here, and so we can see the little red dots indicate that they have the allele for eye color, and then here we have the male. And again, this male has the white dot right there, and then he has a different chromosome that does not contain an allele for eye color, so he has white eyes, whereas this female has red eyes. This is just, again, kind of a diagram explaining what happened. So you have the P generation, so you have your red-eyed female with your white-eyed male. When you cross them, you get the F1 generation. So all of the individuals here have red eyes. Now you cross a female and a male from the F1 generation, and what you find is you get the 3-to-1 red to white eyes. However, it's only the white eyes that are male. So Morgan discovered that there are many genes but only a few chromosomes. So what he deduced from that is that each chromosome must carry a number of genes that come together as sort of a package deal, so that you've got different genes that are all kind of lumped together on the same chromosome. And so what this kind of shows is that certain genes are linked together because they are always inherited on that same chromosome. So linked genes refer to traits that are located on the same chromosome. When you have linked genes, you end up seeing a failure in Mendel's law of independent assortment. And what that means is that sometimes you will have genes that are inherited together. They're not completely assorted independently because it's the chromosomes that assort. And so if two genes are on the same chromosome, you can't separate them. And so you tend to see ratios that mimic monohybrid crosses. So here we're looking at, again, another fruit fly example. And so here we're looking at body color and wing type. And so we've got body color, so white or black, and then you also have vestigial or normal wings. Um, and so what we have here is that the wild type for color and wing type is linked together, and then the recessive color and wing type are linked together. So if they were unlinked, you would expect to see a 1-to-1 to 1-to-1 ratio. If they are unlinked, then the ratios will be altered. So here we have our P generation, and so we have homozygous individuals. So you have a wild type. This is gray with normal wings. And so you can see it's got all the pluses. So B plus, B plus, VG plus, VG plus. And then you've got your double mutant. So the B's are for black, so it's got the black body color, and VG is for vestigial wings. So you can see his wings are much smaller. So now you cross these two, you get an F1 dihybrid, and so now this individual is heterozygous for both body color and wing type, and you do a test cross, which is again when you take that F1 dihybrid and you cross it with another double mutant. So again, this guy's got black body, vestigial wings, and then you look at the offspring of the F2 generation. And so now you've got your expected ratio, so you can calculate, like if you have a certain number of offspring, how many you would expect to be wild type for both, how many you would expect to be black and vestigial, gray and vestigial, and then black and normal. So you can calculate all that out with your expected ratios. But here you have your observed. And so these are going to be the number of individuals that have each of the phenotypes. And so these two phenotypes here correspond to the parental phenotypes. So one is a wild type and one is black as vestigial, so they correspond to these P generation phenotypes. And then you've got these individuals over here, which are a mix. So this guy is gray and vestigial wings, this is black and normal wings. So we call these recombinant phenotypes. They represent a recombination of the genes that we were looking at. So we no longer have wild type body color, wild type wings inherited together, nor do we have the double mutant traits inherited together. They are they've been recombined, so we call them the recombinant phenotypes. This is just a diagram showing how recombinant chromosomes can occur in meiosis. So remember, during meiosis 1, you've got crossing over that occurs, and so what can happen here is when crossing over occurs, you can actually split up the original phenotypes, and that is what happened here with the recombinant chromosomes. And so what you do is, to figure out how often these are becoming recombinants, you take the total number of recombinants, divide it by the total offspring, multiply it by 100, and then you get your percentage. So 17% of the offspring in this case are recombinants. So you have a 17% recombination frequency. So crossing over again breaks up these linkages and it creates new ones. And recombinant offspring formed, they don't match the parental types. So if genes are linked, then independent assortment of traits fails, meaning that you are going to have some traits that are inherited together, and linkage can be considered strong or weak, and that depends on how close the genes are located to each other on the chromosome. So if the genes are on the same chromosome but really far apart, then it's considered a weak linkage, because a lot of times crossing over will split those two genes apart. However, if the two genes are found really close together on the same chromosome, the odds of them being split up through crossing over is much smaller, and so the linkage strength is considered much stronger. So we can use this information to create genetic maps, and what these genetic maps do is they map out a rough estimate of the location of genes relative to one another on a chromosome. And so we will be doing this in class. You use the crossing over frequencies, those recombination frequencies, to help you draw these maps. One map unit is going to be 1% recombination frequency. So if we were looking at this example chromosome and we were looking at these three traits, and we figured out that there was a recombination frequency of 9% between B and CN, and a recombination frequency of 9.5% between CN and VG, but then when we compared the black to vestigial wing, and we discovered there was a 17% recombination rate, we would be able to order these three genes on this chromosome in this order because we know that these two have to be about 9% apart, or 9 map units. We know these two have to be about 9.5, and that leaves about 17 map units between the black and the vestigial genes. So genetic maps have been created for many traits in fruit flies, also in humans and other organisms as well. Here is a gene map for fruit flies again. So your lecture question is, gene maps are only good for genes that are within 50 map units of each other. So think about what that means. If they're 50 map units away from each other, what does that mean the recombination frequency is? And so why do you think gene maps are only good for genes that are within 50 map units from each other? So think about the question. Please make sure you guys send me a message on the class website with your response. All right, thanks.